Hi, everyone. In this explainer, we're going to take a close look at the anti-prion activity of a compound called elacridar, based on the work of Mercer and colleagues. We'll start by establishing it as a potent anti-prion compound. Then we'll move into eliminating some of the established mechanisms you might expect. After that, we'll see how a clue from cell division pointed the research in a new direction, revealing the lysosomal mechanism at its core. We'll then look at how this mechanism also inhibits tau and α-Synuclein aggregation. And finally, we'll wrap up by discussing what this all means for novel therapeutic strategy. So let's dive right in. First up, we'll examine the primary data that initially flagged elacridar as a compound of real interest for treating prion disease. Okay, so what we're looking at here is data from several different marine cell lines, N2A, CAD5, and L929, infected with a couple of different prion strains. And the results were quite consistent across the board. The key metric is the EC50 value. That's the concentration of elacridar needed to cut the levels of the pathogenic prion protein, PrPSc, by half. As you can see, these values are all in the sub-micromolar range, from around 300 to just under 1,000 nanomolar. That's a good indicator of potent activity. But the story gets more interesting. It's not just about reducing the levels of this misfolded protein. Long-term treatment actually managed to permanently cure chronically infected cell lines. You take the compound away, and the infection doesn't come back. That's significant. On top of that, it also prevents the neurotoxicity you see with prions. It was shown to block the retraction of dendritic spines in primary neurons, which is a classic sign of prion damage. And one very important detail emerged here. This protective effect was time-dependent. It worked best when it was applied early on, right around the time of prion exposure. Okay, so the compound is potent. The natural next question is, how does it actually work? The investigation then shifted to figuring out the mechanism of action, and they started, logically, with the most direct possibilities. The first idea was pretty straightforward. Maybe elacridar just gets in the way of the conversion process, directly stopping the normal PrP protein from misfolding into the pathogenic PrPSc form. So to test that hypothesis, they used an assay called RT-Quick. It's a cell-free system that's very good at amplifying even tiny amounts of prion seeding activity. But the results were clear. Elacridar didn't inhibit this process at all, even at high concentrations. This finding argues strongly against the idea that elacridar works by directly binding to the prion proteins and blocking their conversion. All right, so if it's not direct inhibition, what's next? Well, elacridar isn't an unknown compound. It has known molecular targets. It's an inhibitor of drug-efflux pumps like MDR1 and BCRP1, and it also interacts with sigma receptors and an enzyme called ferrochelatase. So the next logical step was to see if any of these known interactions were responsible for its anti-prion effects. And the way they tested this was quite elegant. They used CRISPR-Cas9 to systematically knock out the genes for each of these known targets in the prion-infected cells. The results, as you can see from this table, were completely unambiguous. Getting rid of MDR1, BCRP1, the sigma receptors, or ferrochelatase, none of it made any difference to elacridar's ability to fight prions. So that definitively ruled out all of its known targets. So at this point, the most obvious mechanisms have been crossed off the list. But then, a critical clue emerged from a different set of experiments, ones that looked at what happens when you stop the cells from dividing. And what they found was a really striking difference. In your standard rapidly dividing cell cultures, elacridar worked very well. But if you stopped the cells from dividing, either with a chemical or just by letting them grow to fill the dish, the compound suddenly had no effect. The cells became completely resistant. This was a major hint. It suggests that elacridar isn't just clearing out existing prion aggregates from cells. Instead, it seems to be interfering with the spread of the infection to new daughter cells during proliferation. This observation about cell division was the key that unlocked the next phase. It pushed the researchers to cast a wider, more unbiased net to see what cellular pathways elacridar was actually affecting. And this led to the core finding of the study. To do this, they turned to RNA sequencing. This gives you a snapshot of all the gene activity in the cell, providing a global, unbiased look at the transcriptional changes that the compound induces. And when they analyzed the data, they saw an enrichment for what are called gene ontology, or GO terms. Some of these, related to lipid metabolism, were expected. But then something else emerged that was highly significant, a strong signal for genes involved in regulating cellular pH, and, even more specifically, lysosomal acidification. When they dug deeper, they found that elacridar was causing the upregulation of a whole suite of genes for lysosomal proteins, things like hydrolysis, and crucially, multiple subunits of the VATPase. That's the proton pump that makes the lysosome acidic. So, with this genetic clue in hand, they went to the microscope to see it in action. First, because elacridar is what's known as a cationic amphiphilic drug, or CAD, it naturally accumulates in acidic places inside the cell. Sure enough, they saw it co-localizing with markers for functional lysosomes. But here's the key finding. Treating the cells with elacridar actually increased the intensity of these markers. This means it wasn't just sitting in the lysosome. It was actively enhancing its function, making it more acidic and better at breaking things down. And this is a distinct mechanism. It works differently from other similar drugs like quinacrine because it doesn't cause phospholipidosis and it acts independently of TFEB, which is the main transcription factor that controls lysosome creation. Okay, so the mechanism is lysosomal enhancement. Now, since lysosomes are the cell's main recycling center for all sorts of protein aggregates, not just prions, the obvious next question was, could this be a more general strategy? So they expanded the investigation beyond prions. To test this, they used special biosensor cell lines that report on the seeded aggregation of either α-Synuclein or Tau. They then exposed these cells to brain material from mouse models of relevant diseases, one for α-Synuclein and one for Tau. And the results were positive. In both cases, elacridar treatment significantly reduced the formation of these aggregates. This is strong evidence that enhancing lysosomal function isn't just an anti-prion strategy, but could be a broadly applicable approach for a range of protein misfolding diseases. All right, let's pull all of this together. The final step is to synthesize these findings into a coherent model and think about what this means for potential clinical applications. So here's the proposed model. Elacridar boosts the lysosome's ability to degrade proteins, likely by increasing its acidity. This helps the cell clear out newly formed nascent protein aggregates before they can really take hold and establish a chronic state. Now, remember how it works best in dividing cells and during the initial phase of infection? That really defines its therapeutic niche. It suggests it's likely not going to be effective for treating someone with advanced, established disease. Instead, the real potential here is as a preventative therapy. You could imagine giving it to individuals with known genetic mutations that put them at high risk, administering it before symptoms appear to prevent that initial accumulation of aggregates.
So what this work ultimately does is uncover a novel TFEB-independent pathway for activating lysosomes. And that leaves us with a really important question for the field to tackle next. What other pathways like this exist? If we can find other ways to modulate lysosomal function that don't rely on the master TFEB switch, we could open up entirely new avenues for developing therapies against a whole host of neurodegenerative diseases.